let me actually show you this. Scroll, or I could just use the mouse. That's not nothing. Well, let me start by showing you a very typical data analysis scenario if you are a biologist, right, or people who are enjoying the genome science. Right? So you have collected some clinical samples, and then you have heard about the great technology, the sequencing, microarrays, proteomics, whatever you have. And then you have known about the available bioinformaticians on campus, and especially coming from Allele's group. You perform analysis through a standard pipeline, and then you get to a so-called upstream analysis that give you very nicely written report with numbers and genes and their statistical significance. But is the job of bioinformatics done? And I would say that I'm trying to convince you in the next 20 minutes or so that the job has really just started. And the actual bioinformatics work is actually trying to bridge the gap between the analyzed data from the pipeline and the downstream analysis that you have to do back into the wet lab and the clinic. And this is what we call the modeling part. And it turns out that nowadays you cannot automate the modeling part. And there's lots of tools available, and there are lots of just a complexity inherent in capturing the data to build your disease-specific model, and that makes actually bioinformatics both exciting and challenging. All right, so I'll just tell you one example of something that you may not be aware of. You're familiar with the lack of false negatives in genomics, but if you are venturing into metabolomics or proteomics, you know that you often see, even in the highest sensitive instrument, the so-called abundant protein, the structural proteins, because they exist in sometimes 10 to the 8th power level of abundance than some of the proteins that is considered as kinases, receptors, or even signaling molecules. And our instruments sometimes are so incapable of capturing those signals, so unless you understand how to use the bioinformatics and use statistics to overcome that, then you're really going to draw the wrong conclusion. All right, so maybe there's nothing, but there is actually, the signal is there. And other issues could be that there's a lot of variability in your data. So a very typical analysis report you get may look like this, but somebody have done the research to show that even for the same individual, right, if you look at the correlation variance within the same individual, if you measure it multiple times, you get lots of variability. If you measure it in the group setting, and the variability is higher. And sometimes this variability can even outweigh the sensitivity of your instrument or the statistical fold change that you collect from your pipeline analysis. So if you don't understand that, you just use your own data set without regard to everything that is the big data that's out there, you may actually draw completely wrong conclusions. And that's, I think, why some of the statisticians actually have written a report and said many thousands of the NIH funded papers and the conclusion may not hold. And there was actually 2 years ago, there was a splash on a paper like that. And then sometimes you will see the very small changes depending on which instrumental platform. I'm now going to elaborate on that. So there's the certainty of death and small chance of success, right? But as bioinformaticians, obviously, we like these kind of challenges. We like to be able to help. And as bioinformaticians, we don't think about the problems at the level, purely at the level of molecular measurements, right? For true that most of the bioinformatics measurements are at the gene level. For example, if you want to get the genome sequence, you're measuring the mutations or SNPs. Or if you're doing proteomic study, you're trying to measure protein, the diversity of proteins found, peptides, and their abundance level. But as bioinformaticians, we're starting to think about more abstract levels and things that are more complex, right? And things like if you put the metabolites together into reactions, what does the reaction look like? If you put two proteins together, do they bind? Do they actually form complex? Do they actually modify each other? And then if you have groups of proteins, do they form modules that are closely associated as protein complexes? And does these modules form biological pathways? And these pathways, do they actually coordinate into a function of some sort? And then all the way to something that is so complex, so abstract, what we call the networks. Right? And the network are not as messy as the big data because they certainly have smaller volume, but they are certainly a lot more complex and abstract than the big data because at least for the data, you understand this gene has this level of expression, but for the network, and this is completely outside of the realm of a traditional biologist. But this is where we thrive in. So I call this a hierarchical biosystems perspective. And these skills actually will extend from the molecular system level in the more translational aspect of informatics that connect with the clinical informatics, which is at the other spectrum of the informatics. The Informatics Institute director, Jim Cimino, is an expert on the clinical informatics. So what is the difference between clinical informatics and bioinformatics? So at the very obvious level, you know that we're working on completely different data type, right? So in clinical informatics, they're working on clinical records, lab tests, prescriptions, imaging data, literature, and physician notes. In bioinformatics, we work on genes, molecules, reactions, pathway, and network. But there's a continuum, as illustrated by this figure. If we're working on a very complex different components and systems, and from these genes, and we can actually get the association to the diseases. And these diseases actually are associated with populations. Right? So if you take that perspective, actually there is a way to bridge between bioinformatics and the clinical informatics. And I think that there's lots of institutional wisdom to actually, I think that's part of the reason I joined here, to create this a single institute where these two ends of the spectrum meet. Right? And you don't usually find institutions that are created this way because of the historical reasons primarily. Right? And if you think about the translational bioinformatics, which I pride myself to be living in, I also want to distinguish or also help you distinguish that it's not a canonical bioinformatics and it's also not applied bioinformatics. So according to the NIH consensus definition of translational work, you have to have 3 degrees of translation. One is moving from model organism to humans. Another is moving from population to individual patients. And the third is moving from the research lab to healthcare applications or drug discovery settings and reduce it to practice. So if you think about the 3 bioinformatics, right, so at the canonical bioinformatics level, if you think about it, primarily we're doing multivariate analysis, but still treating variables as if they're independent of each other. We're not trying to look at the relationship per se. In translational bioinformatics, since we're dealing with complex systems, we have to model those in. And then in canonical bioinformatics, the scope of analysis is primarily on the lab data that's presented, and then you analyze it per se. But translational bioinformatics, we have to increasingly deal with the related public data set. We're not happy with just analyzing your data. We have to actually bring in relevant data. You may say, oh no, that's not the specific subtype of the disease. But there are actually lots of black holes, as we often see in your data. Unless we bring in those complementary data set in a broader perspective, we actually are pretty weak in analyzing your data because we're not as smart as you are, not as well trained as you are. You all know the individual functions of the genes, but as bioinformaticians, we lack those domain training. We have to use these the public data set to overcome some of that. But along with that, we probably can eliminate some of the biases. And then there's the heterogeneity of data type. In canonical bioinformatics, you're dealing with maybe just the genomics data, but in translational bioinformatics, we have to bring in so many data types from public databases, and that's make it more challenging and exciting. And at the patient's level, I think at canonical bioinformatics, mostly we're dealing with cohort data, right? But translational bioinformatics, for the nature, you have to engage on clinicians and challenge them to give you individual level data so you can actually map out the individual heterogeneity. And then the collaboration, it's much broader, and the primary interest is on the biomarkers drug instead of the algorithm and the knowledge that you traditionally want in the canonical bioinformatics. But on the other hand, the applied bioinformatics, I would say, is more pragmatic, right? It's whatever is available, and usually the people in charge is not a bioinformatician, and the bioinformatician is serving as a service mode, which is entirely okay, but I think of translational bioinformatics make this more of a dynamic balance between the domain expert and the bioinformaticians. And if I haven't bored you enough, and I can also tell you that in bioinformatics, we're trying to move into a more systems perspective. Right? So if I convince you that it's all about the hierarchy and the network is sitting at the top of the hierarchy, and there's special interest within the bioinformatics community to study that, and then you know that we're interested in studying the system structures, and essentially everything that exists and their relationship form the structure. And dynamics, what we call the dynamics, what you may call it as a temporal analysis of expression data. How does a system or things change over time? And even before disease and after disease, or early stage of disease, or different staging of the diseases. And system control is essentially finding maybe the drug target that essentially if you change it, and it will reverse the disease to the normal conditions. And what are those control elements? It may be proteins, maybe RNAs, or maybe other things. And the system design is really started with the synthetic biology community, where we're trying to change it, come up with a new therapy. So, I mean, the things that the translational biomedical researchers are interested in, we as bioinformaticians also want to participate, but more from the in silico point of view. Right? And these are some examples. For example, determining all the structure of genes and chromosome structures. These are part of the system structure. If you have all the differential equations in the microbial system, And if you want to say what is the change in concentration of glucose to certain phenotype that you want to measure, if you have all of that reaction constant, you can simulate it in the in silico setting. So that's what we call the systems dynamics. System control, you draw a diagram based on your the experimental data, and in the abstract diagram that look like this, and one gene stimulate another gene and one inhibit another. Once you have this diagram, you can actually clearly pinpoint how am I going to actually block certain target to achieve the desired effect. Then the system design is more interesting. Can you actually design a system that not only change one thing, but probably change a couple of things to achieve, in this specific case, an oscillator function that involves the changing of two genes introduced into the organism. Okay. So I think if you ask us, what do you do as a bioinformatician? I can't really tell you in one sentence, because according to the two dimensions that I just talked to you about, one dimension being the level of granularity from molecules to populations, another being the different aspects of the system sciences. Depending on where you are in posing those questions, and I would give you a completely different answer, right? So more traditionally, sequencing, sequence comparison, multiple sequence alignment, they fall into molecules and structure space. But as you can see, that these things have morphed and diversified into such a complex repertoire of skills. And I believe that I look at the UAB is known for immunology and the cancer immunotherapy. And this is actually more challenging for bioinformaticians, right? Because you're thinking about intracellular models that you have to model multiple systems at the same time, and then you're thinking about the control. What are the regulatory models that control the development, cancer metastasis, immune response, and then the therapy as a design. But then some of the things that are more mature are written in here. Right? And then there are things that bioinformaticians are limited, but luckily we have epidemiologists studying at the population level, and then some interface with bioengineers. I'm hoping that to engage them more in the future, and then they're really good at modeling at the cellular level and above. Right? So, well, I think it's pretty complex, and I think you may ask, well, is it really tricky to work with the computer geeks, right? Because I don't speak your language, and I don't even know how much my work would cost in terms of the time or money, and how do I approach you, right? I think I want to pose a model, and if probably I don't have time for a concrete example, but this model, I'm going to actually ask you to memorize it, and hopefully you know that there are certain processes involved before you can do this fun stuff, which is the part of storytelling. Right? As a biologist, you would like to go to a public forum in Science or Nature or professional meetings and talk about the greatest inventions. But those things have to be connected from the massive big data that start with the bio-IT programmers. Right? This morning, Bob and I were talking about how to name these people, and then I think that they're more bio-IT specialists and programmers because their task is to marshal the big data through some pipelines to the point that they are reduced. And then the biostatisticians are very important because then they help to assign the statistical significance, although there's overlap. And then after the data are reduced, we need the bioinformatics data analyst to really bring in the external data and then bring in the custom analysis capability and iterate through this process. And this is a more time-consuming part. And this is a part that I hope, if you want to invest in your research, this is a part that I hope that you understand this could be 10 times more costly than some of the standard analysis upstream. But I think it is our goal at the Informatics Institute and the Allele to actually coordinate this process so that more and more of the research will become more common commodity and fall into the pipeline. So the first part will become more and more easy, and then this part you can do it faster and faster. Right? And this part you ask the questions, you visualize the data, you explore the data together, analyze it. It has to take the collaborative team of bioinformaticians and biomedical researchers. And to the point that when you tell the stories, and then everything will be really nice, and you get the grants and papers and everything. But let me actually just stop here and see if there's any questions before we move on to the second part. I did have some example, but I don't think I'll have time to go through it. Any questions?
Jake, perhaps you could give us just a little bit more sense of what you mean by data wrangling. It sounds like it's a wrestling match. What's involved in data wrangling?
Right, and the data from different sources usually come in different formats. They are unstructured to begin with, so part of the work is to converting the data from one format to a more standard computer readable format. Either it's in the relational database format or XML format. But the more challenging part is to really standardize the semantics of the data. For example, a number 16 could mean the age or it could mean the reading. But sometimes people would have overlapping name, or even they use the two different systems, they use the same name, but then different meaning. And even for age, in China, if you go to rural places, they will start counting the age, the person born to the age 2 already. They count the prenatal ages. So if you have a system that's incompatible, it's going to be very challenging. Data wrangling capture all of that, just a very tedious but very challenging, very important task upfront.
So is it fair to say, even if someone is not a bio-IT programmer, understanding the data and what it means also involves the investigator at that stage? Because some of those examples you just gave might be known to the investigator who collected the data, but it may not be immediately apparent to the bio-IT person.
Exactly. And in fact, the earlier the better. And one of the interesting story is before I leave IU, I maybe overcommitted myself to working with a clinician at IU, and I thought it was a very straightforward analysis, so I directly gave the analysis to my student, and then the student did analysis, and we came back, and then we were trying to analyze the data, and all of a sudden the clinician said, oh, by the way, the mass spec people didn't do the experiment right. They were using the wrong controls. And then if I were actually engaged early on, I would look at it, I would challenge it, and I think we wouldn't have created all these waste and frustration. But it happens. All right, so we'll have more questions at the end. Maybe, let me welcome Elliot.
I'll just do that.
Okay.
This is my
So what I want to do is try to answer the question, how can you make bioinformatics work for you? That is, how can you get access to the services, resources, expertise that can allow you to take your data working with a bioinformatician and get that data analyzed to answer the various questions that you may have. And so what I'm going to talk about is the bioinformatics services available through the CCTS Informatics component. But I want to emphasize that what's available is not solely within the CCTS Informatics group. That is, we try to collaborate heavily to get expertise from different sources, both within UAB, but also within the CCTS partnership, such that we can find the expertise you need if we don't necessarily have the expertise in-house. So to start off, as you may know, the CCTS is a partnership of institutions around the Southeast. And a number of those partners that have various levels of informatics expertise have gotten together to form the Southeast Informatics Consortium. And that consortium works together to identify resources available at one of these partner institutions that might be able to support a project, a grant collaboration, research support, education that's available from one partnership but not available from another. And so we can help identify where you can go to get the optimum support for whatever it is you're looking for. So if you go to the CCTS Informatics homepage, and a quick way to get access to that is uab.edu/informatics. That will take you to the CCTS Informatics homepage. And then there's a tab there for partners. And that will give you the list of experts who head up some of the informatics cores or have the expertise at a number of these institutions. There's also a spreadsheet you can download that has a list of experts and expertise. Now, you don't have to do that if you don't want, because you can come in just to CCTS Informatics, and if we see that you have a particular problem that needs the expertise of one of these people or one of these resources here, we'll put you in touch with them. But right now we have a number of ongoing collaborations. We've collaborated on grant submissions, certainly research support. It's been a very active consortium. Now, if we come to UAB, at UAB there are also a number of resources available in different units around campus. And the same thing holds. We are aware of who these people are. We work closely with them, and we try to connect you when there's a problem that we can't solve, but they may have the expertise to solve. So in addition to CCTS Informatics, this includes the informatics facility at the Center for AIDS Research, the Heflin Center for Genetics. David Crossman is the bioinformatician there, and a lot of people work with David to analyze sequence data that's been generated at the Heflin Center. The section on statistical genetics in the School of Public Health, Department of Biostatistics, has a number of experts in applications of statistical approaches to analyze biological data. And that really crosses over a lot with the bioinformatics that we do. So we work closely with them, again, to provide these resources. And then finally, basic resources like the computational facility, the computers, high-performance computing that's needed to do the analysis that we're working on. We can help you get access to those resources and utilize those resources in order to support your analytical needs. Now, within CCTS Informatics, we do provide a number of different types of consulting services, from traditional bioinformatics, next-generation sequence analysis, analysis of microbiome or shotgun metagenomic data. Last forum, Jim Semino talked about clinical informatics. So today I am concentrating sort of on the analysis of basic research data when I talk about bioinformatics, but we also help support some of the clinical informatics data access and processing, as Jim and Jeff Gordon and Matt Wyatt described the last forum. And then we also provide services that will develop a number of different tools that might be needed in order to analyze data. And then a strong component, a heavy emphasis, is on education and training. And all of this is done in concert with all the partners. And the one thing I should emphasize that wasn't on that partnership, because I see it as an overarching entity, is the Informatics Institute itself. So the formation of the Institute really provides an umbrella under which all of this can much more easily happen. these days when people need different types of analytical services. Within CCTS Informatics, we have 5 full-time bioinformaticians and several programmer analysts that can help you with your work. But importantly, and really the bottom line from everything I'm going to talk about today is how you can get access to our services and resources. So Research Commons is the basic place to start to get assistance. So you can go to the CCTS website, either on its homepage or go to the Research Commons link here. That gives you access to the phone or email. So anybody requesting help through that mechanism will be directed at the most likely CCTS component to provide assistance. So anything to do with bioinformatics or clinical informatics data access will come to the informatics group. And then what we do when you do make initial contact with us is there may be an initial consultation in which we'll talk about the research, talk about the analytical problems that you have, and talk about how we might go forward in helping you solve those problems. We can help on a regular basis with grant preparation, publications. All of this assistance is available by starting with the Research Commons. So what I want to do now is just spend just a brief period of time just giving you a little bit of a taste of some of the types of bioinformatics analytical problems that we deal with. I'm not going to go into any detail. Some of these are early projects we're working on. Some of the projects, some of the screenshots I'm showing you have been published. I'm not mentioning any names in this. I'm just showing pictures of some of the results of what we do. But what it should do is give you a feel for some of the types of analytical services that are available. So one is genomic, so starting with sequence data. So as an example, if you're interested in human genomics and you want to look for rare genotypic variants that might map to a particular phenotype, we can help you analyze, look for the variants that are in those data sets, and then start to mine that data to see if there might be any signal there that would be important. So you start to look at the sequences here, and this is just a representation of 2 individuals having their sequence compared to the human genome reference, and the little vertical bars here are areas where you might have heterozygous positions or positions that are variants with respect to the human reference genome. This is just to zoom in here to see a particular variant at one position there. So the key there is to analyze all this variant information and turn it into data that might be useful. In this case, data that looks at individual genes, looks at the variants in those genes, and tries to make a prediction as to how impactful that variant be in terms of its association with a particular phenotype. So we do also a lot of work with microbial data, viruses and bacteria. So a similar type of thing is done with viral genomes. And this is not sequencing human genomes, but viral genomes. But again, looking at the same thing, looking at allelic variants with respect to reference genomes and asking the question, is there association between particular variants and a particular phenotype of the virus? It might be disease, it might be response to drugs, whatever it may be, we can assist you in making those associations. This is something that comes out of what I call traditional old school bioinformatics. This is a project we worked on in which we were analyzing the evolutionary relationship of hepatitis C viral sequences that were isolated from a variety of patients. And so this depicts the variant, the differences between those viruses displayed on a phylogenetic tree. But you look at this and you also get a lot of other information in addition to how much any one virus varies from another. The topology of this tree, the fact that you have this bush-like structure, tells you something about the epidemiology of the virus. It tells you that there's a bottleneck to the transmission of the virus from one individual to another. That is, there's a single genotype that's able to be transmitted between individuals, but once the individual gets infected, then the viral sequence space expands and the virus can mutate to a great extent. But you always come to this bottleneck for transmission. And that has impact on development of drugs, development of approaches to preventing hepatitis C. RNA sequence analysis. So looking at transcriptome differences between different samples is something that we've done a fair amount of work on. As I mentioned before, microbiome and metagenomic analysis. So if you've read either the scientific literature or the popular literature, you know that the evidence that points to disease associations between the bacteria and viruses that inhabit your body, how those and changes in that population can impact disease, can impact all sorts of phenotypic conditions, is really just starting to be appreciated. And so using microbiome and metagenomic analysis can help get at that. And the UAB microbiome facility is able to process the samples, from taking the samples themselves, processing them for sequencing, sequencing them at the Heflin Center, and then CCTS Informatics analyzing that data for you. And so we get information on the distribution of the different bugs that happen to be in a particular sample. We can compare groups of bugs to ask a question whether or not there are differences in the patterns of microbes that are present in different groups of samples. So down on the left is a principal coordinate analysis that tells us we have a grouping of the orange samples together and the blue samples together, and they actually have meaning in terms of the patients from which those samples were derived. But we can, and one of the things that we do to better, so we run thousands of microbiome samples a year here at UAB, and you can't analyze that by hand. So we develop a pipeline in order to automate that part of it. So that's the easy part. You have the pipeline, it analyzes the data, then we try to work one-on-one with you in order to derive meaning from that data. Metagenomics is something that we're also getting into, in which you have a lot more sequence information, but you can have a lot more attention played to individual variation between the different bugs in a sample, and you can trace individual isolates, individual bacteria through different samples. And so look at patterns of transmission for different studies that you might be involved in. A lot we've done in terms of bacterial genomics. We probably worked on sequencing several dozen different bacterial genomes, the assembly of those sequences, and again, looking at variants in the sequences to see if they might match to some phenotypes that would be important to disease. This is one project on Mycoplasma Genomics where we helped assemble the sequences of over a dozen different Mycoplasma genomes and then looked at variants in that setting. So finally, we also try to provide tools, and there's some tools we provide that we develop in-house. There's a tool to support education called Kaizen that's been developed along with James Willig. Other tools that we develop that are available for your use, or we can talk to you about it if you have such a need. But if you do have students, postdocs, or yourself, and if you'd like to do some of the next-gen sequence analysis, we also try to provide a platform from which you can do a little bit of that yourself. Galaxy is a web-based platform that provides access to next-gen sequence analysis tools. So we can work with you to help you use those tools to analyze your own data sets, or if you prefer, like for the other projects I showed you, we can analyze your data sets as well. So I want to stop there in terms of what we do, but I do want to mention a few upcoming events that will be occurring over the next year. In September, on September 14th, and specifically, we're going to have a metagenomic workshop in which we go into the uses of metagenomic data, how you process that data, how you understand the results from that data. The next day, on the 15th, Claire Fraser, who's the Director of Institute for Genomic Sciences from the University of Maryland, she was Director of TIGER, the Institute for Genomic Research, that sequenced the first bacterial genome. She'll be coming to give a talk on the 15th. And then we're also going to be providing next fall a Galaxy workshop to give everybody more knowledge about how you utilize the tools that are part of Galaxy. So I'll stop there and answer any questions anyone may have.
So Elliot, in the broad range of different applications of analytics and informatics, one thing, And I don't know whether this is for you and Jake, or whether it's actually for Jim. I think Jim was on an airplane and was unable to join us. How about the analysis of images? Because I think for some of us who are here this evening, image analysis is important. How Jake or Elliot, how should we think about that? How could we think about that?
Well, one thing I'll point, so that's an example of something we don't have direct expertise with, but there are people on campus. For example, there's a number of different groups that utilize the high-performance computing resources on campus to analyze their data set. One of those groups is a group that works on images and utilize MRI scans and such. And so depending upon the types of images you have, we can help connect you with people who have that particular expertise.
So from disciplinary point of view, the imaging analysis has been morphing into imaging informatics. And then it's a very hot area because traditionally, if you just talk about image analysis, it's about recognizing patterns within the image and comparing features and annotate features. Those are within the realm of computer science and some overlap with biomedical engineering. There's not much, but I think the computer needs to improve, and therefore additional layers of data are being added. For example, correlating the brain structure with the functional MRI data that requires some annotational information of the images. So that goes beyond the computer science area. And now I think that people are very interested in correlating these images with the genomic markers. And these multimodal analysis, then that's actually an extension of translational bioinformatics. If you're just talking about images, of course, the image will be pulled from clinical records. So I would say this is an area that we don't have I wouldn't say we don't, but within the Informatics Institute, we don't have expertise. But at some point, if we have that need and want to lead forward, we need to hire specialists.
We do have a lot of the computing science department. My former supervisor, Tracy Zhang, in the Knowledge Discovery and Data Mining Lab, does image analysis. So that probably is something to talk to. And she does video as well, other mobile stuff.
John, do you know what is being done at Auburn?
No. Not entitled image analysis.
Adrian, do you know what's being done at Auburn?
There's a certain amount of people are doing some, more analytical processing, but it's still limited. And that has been a big focus of the retreat we had, was to try. Last year we spoke a lot about this, how to different methods. But I feel like we need to move at a different level.
Sure.
We are trying doing, but with the tools we have, it's not working anymore. Because we are merging so many different modalities and genomic information is getting too big, and so that I really need to see what else can be done.
So I actually, with the question, I was setting up an opportunity because with the imaging capacity at Auburn, which is a higher resolution MR, and of course that's not quite pathology, but it's another kind of image. There's interest in developing protocols, a number of different imaging protocols, and then the question, of course, is how do you deal with that information beyond what I would do, which is I look at it. I'd like to think actually I have a fairly sophisticated image processor that's looking at those pictures, but it obviously also has some limitations. But that might be something that we would want to be thinking about forward-looking, because I think the folks at Auburn, if my instincts are correct, at least with MRI-type imaging, they're not only engineers interested in producing the images, but they're engineers and data scientists interested in analyzing them, what they produce. So that might be a forward-looking thing, and it would serve a number of projects which are going on campus in terms of really whole body. The 7T at Auburn is whole body building. So it's not just neurologic, though. There's a lot of interest in that. Okay, quite interesting. But there's a lot of interest in neurologic imaging, in cervical spine imaging. But it's a huge opportunity for us to be thinking about it and go forward with. It may well be is a plug. Auburn is trying to do some, what do they call it, group recruitments. And they're looking to recruit people with interest in informatics and image analysis. So there may be a way to work with them, which works for everybody, I guess, the bottom line.
And certainly, UAB has just acquired, one of the most, top-notch imaging facilities in terms not only of fMRI, we have a scanner, but also in terms of the PET MR. I mean, we have within a few years, we've become like the top place in terms of imaging, and certainly with 7T. So that is, I think, great. We all work with each other. I mean, I scan at Auburn on 7T, so that's a huge opportunity.
And so again, we might think about part of that solution being from within UAB, but we may also think a little bit more broadly in terms of the solution. There will never be a single solution, but the progress in moving forward to image analysis, I would wager that the guys at Auburn would be very interested in working on this, and it's a way to sort of leverage what we got and what we want to do. Elliot, if someone has is going to be generating genetic or genomic data, one of the things that I think we've all learned is these machines, which are very nice, spit out information at a rate which is rather phenomenal. How should we think about setting up the analytics of that in advance of putting the first sample into the machine?
The most important thing that you can do is come and talk to the people who are going to support the analysis before starting. And that can easily be us. It might be, if you're using a Heflin Center, some of the people in the Heflin Center. But we can provide a number of recommendations on how to proceed, depending upon what your ultimate goals are. Because there are different approaches to doing the sequencing that can have very profound effects in the ability to accomplish the sequence assembly and variant discovery and such down the road. And you have to take that into consideration up front.
Okay, so then at least one of the ways for investigators to think is to come to perhaps to Research Commons, develop, at least have that initial conversation. If you're in an RNA-seq space, it would be understanding read depth, what you can learn, what you can't learn, to your point, major signals, minor signals, that kind of thing. Is that fair?
Yes, definitely. And if you're interested in assembling genomic information, genomic chromosomes from whatever organism, how you assemble them, the extent to which you want to complete the genomes, your ability to do that will vary depending upon the decisions you make up front in terms of the sequencing. So that is another area where it's very important to get it right.
Other questions? Thoughts, comments? Guys, it's 6 o'clock. I want to thank you both very much for being with us tonight. We've had some come in over the web, some of us here, there's still lots of food and plenty of beer. I'd like to thank you